Hi, I'm going to go over this genetic engineering gizmo with you. Before we do that, I just want to give you a quick overview of genetic engineering. Hopefully you saw this slideshow, but you can see that these mice have been genetically engineered to glow. Any kind of modification of an organism's genome is what we call genetic engineering. And when you modify their genes, you affect the proteins that they make. And when you affect the proteins that are made, that affects the overall trait of the organism. These are different ways you can modify an organism's genes. Scientists love to modify bacteria because they're so simple. And then those bacteria can then produce the protein that you want. So it could be insulin, that they're producing when you put the gene in to make insulin, or it could be plastics or even fuel. They can engineer plants now to produce proteins that will kill pests so that they're resistant to pests, or resistant to drought or harsher weather. Most of the crops that we have in the United States are genetically modified. And here's a couple more examples. Here are reasons why they modify crops. So these are all so that farmers can have a better yield and produce better crops for people to buy. Animals can be modified as well. So here's that glowing mouse, but they can use it to track proteins. They can use it to produce more growth hormone so that the animal can grow bigger and faster, produce less smelly cows. Here's a web producing gene that I don't know why you'd want spider silk in your goat's milk, but there we have it. And then here are some concerns that some people have about genetically modified organisms. For example, if a genetically modified organism drifts over and then interacts with another non-genetically modified organism. And also some, questions about, at what point is it unethical to modify something. But most of the crops that we have in the United States are genetically modified and allow us to have the food that we have and at the prices that we have. So here is a lab that's going to show how we can make a genetically modified corn plant that is resistant to these caterpillars, this type of pest. So here is, you can answer these questions, and here are 12 plates of bacteria and different plates of bacteria, and on them they have the same kind of caterpillar that loves to eat these corn plants. And what we're going to do is we're going to see which of these bacteria produces a toxin that kills the caterpillar. So which of these bacteria is kind of naturally toxic to these caterpillars? So we're going to go ahead and push play. And we can see that this bacteria naturally will produce something that kills these caterpillars, and this one as well. These are somewhat toxic, but it doesn't kill all of them. So we're going to go ahead and pick one of these petri dishes, and we're going to see if we can isolate the gene that is producing that protein that's toxic to the caterpillars. So here are all the genes that are in that particular bacterium, and we'll test them one at a time. We'll take gene A and we'll put them in this bacteria, gene B, and gene C, and we'll see, because we're trying to figure out which gene is it that we're going to take and put in the corn plant. So we'll go ahead and push play. Okay, so it's none of those. So we'll try gene D, E, and F. Let's go ahead and push play. So E is the one that is producing a protein that's poisonous to the caterpillar. So I'm going to go ahead and pick gene E, and we're going to insert that into our corn DNA. So here we need to figure out which promoter we want to use, because different promoters are going to show up in different parts of the plant. And we want a promoter that's going to show up in the corn kernels and the leaves and the stalks. We want our gene to go in all of those places because that's where the caterpillar is going to target. So when we turn the lights off, we can see where those promoters are because they have been given a gene that also allows them to fluoresce or glow. Okay, so this plant looks like it's got the promoter in all of the parts of the plant. This one is missing the roots. This one's missing something. I can't really tell. Is it the leaves? And this one's missing the roots. So we're going to go ahead and go with promoter 1 because we want the gene to go in all of those places. And then this shows where the gene has been transformed or inserted. So this is a callus is a group of plant cells, and they put the gene right here. This blue part is the gene. Now, one thing that you'll notice as you read this is that so we already picked that part right here. So the genes are the green bars, and we don't want, okay, so the new gene is our blue bar, but problems occur if the new gene is inserted in the middle of an existing corn gene, a green bar. So in other words, we don't want our blue gene, the new gene, to be in the middle of a green gene. So do you see how this blue gene is in the middle of the green gene? So that is not a good, that's not going to make a successful plant. There's problems that happen. Here is another of our new gene from the bacterium, but it's in the green area, so we don't like that. Again, here it is, that's a problem. Okay, here it's not in the green gene, so this one looks like it could grow properly, number 4, and number 5 looks like it could be okay too. So I'm going to pick 4, and then I'm going to click continue. Okay, and we're going to go ahead and just grow it to see if everything's good. Okay, so we can double check. And this is a normal plant without the genetic, it's not genetically modified, it doesn't have the gene from the bacterium. Leaves look good, corn looks good, roots look good. Okay, here's one that's been transformed that has a new gene. Leaves look good, corn looks good, and roots look good. If we were to have put, let's say, one of these where the new gene is in the middle of another gene, let's see what problem would have happened. So here, leaf looks good. Corn, do you see how there's a problem with this corn? I think that might be better because you get 2, but something's wrong with that. And then the roots look okay. But something was wrong. So we're going to go and make sure that we pick a callus where the new gene is not in the middle of another gene. And we're going to go ahead and grow it. We did grow it. Everything looks good. Okay. And then we're going to, now we're going to go ahead and we're going to add the larvae, those caterpillars that love to eat corn. Okay, so we're going to add it, and we're going to grow both plants. So we know that a normal plant is going to get eaten up by these larvae, but But what we hope is that this genetically modified plant that now has the gene from the bacteria that kills those larvae, that this one is not going to get eaten up. So let's see what happens. And if we go close up, we can see the leaves got eaten up, the corn got eaten up. The roots look okay. It's hard to really see, but they look okay. The transformed plant looks good. Corn looks okay. Roots are okay. And so we're going to go ahead and show the statistics. And you can see in this graph that the transformed plant only had 2 caterpillars found on them, whereas the control plant that was not genetically modified had 74 caterpillars on it. And that's why the control plant only ended up yielding 8% of its corn, whereas the genetically modified one had a 99% yield. So only like a little bit of its corn was damaged. Let's go ahead and submit it for review, which is what this part E says. And it says, congratulations, you have created a corn plant that is resistant to whatever this bacterial name, insect name is. And now we're going to do test 2. Okay, so we've successfully created a genetically modified plant. So let's go ahead and do test 2. This one I'm going to go a little faster just so you can see an overview, but you'll be able to get the gist of it. So here you're going to create a genetically modified corn plant that's resistant to a larva, another larva, but it's beetle grubs. So it's not those caterpillars, but these are beetles, and these feed on the roots. So first we're going to just do our resistant test. We have to find out which strain of bacteria is going to kill these beetles. Nice. So we've got 6 or 11 for us to test. I'll go ahead and pick 6. Okay. Now in bacteria number 6, we've got all of these genes. So we need to figure out which gene is the one that's producing the protein that's toxic to the beetle grub. So I'll go ahead and push play. Okay. So it's not A, B, or C. We'll try gene D, E, F, O. Okay, so F is the one that is that we're going to put into our corn plant. So we're going to click F, click continue, and now we have to figure out which promoter do we want to use. We want to use a promoter that is going to put the gene in the roots, because that's where the beetle grub feeds. Okay, so I guess, I don't know if it matters, but we could put it in either this one or this one because it's the root. I'm going to just pick this one. All right, so now we have to figure out which callus we want to use. And remember, we want to have our green bar not be in the middle of, our blue bar not be in the middle of any of the green genes. So that one's a problem. This one is nice and by itself. So number 2 could be a possibility. Number 3 could be a possibility. Number 4, no. And number 5, no. So let's go ahead and go back to 3. So 3 is this blue gene. It's not breaking up any green genes. So let's go ahead and say continue. Let's see if our plant grows correctly first. And we'll check. Everything looks good. So our control plant should be okay. Let's look at our transform plant. No oddities. Okay. Now we're going to add the larva. So we're going to go back. We're going to add these beetles and see if our genetically modified plant survives the beetles, kills the beetles, basically. Oof. Okay, so you can see the beetles really messed up the growth of this plant. There they are. Okay. So when you're messing up the roots, then the plant's not going to grow correctly. This one grew a lot taller and is beetle free. So let's take a look at our statistics. Yes, we've had a successful corn yield with our genetically modified plant. Barely any beetles. Let's submit it for review. You create a corn plant that is resistant, and then you can do the last task. So I'm not going to do this one, but here is you're going to be creating a corn plant that is resistant to herbicide. So when you spray corn plants with herbicide, you don't want the corn plants to die, you just want the weeds to die. So you want your corn plant to not die from the weed killer. So this one is going to be a little bit different, but the same idea. And instead of adding the pest, you're going to be adding the herbicide. So let me know if you have any questions. Otherwise, good luck and have fun.